Okay, so this is 3.5.1 photosynthesis. We're going to talk about the light-dependent reaction. This is the first module in Year 2 biology, depending on what example we're looking at, but in AQA, this will be the first section of that. So what we've got to do to begin with is remember the GCSE equation for photosynthesis, which would be carbon dioxide plus water gives us glucose plus oxygen. That's when we have light in the presence of chlorophyll to trap the light. I'm going to talk about how that's different. So if we balance that up, remember from GCSE, it would look like this. Now, photosynthesis happens in the leaves, but specifically in the chloroplasts of the leaves. So the chloroplasts contain chlorophyll, and that's about as far as we got at GCSE. But at A-level, we have to understand that we have internal structures that allow different parts of the reaction to take place. So we have a green fluid in the inside here, so all of this space here, called the stroma. And that's where the next part of the reaction is going to take place, the light-independent reaction. Now, in the middle here, we have these structures called grana. So this is one individual granum. And if we look at this and this and this, we have grana, which is the plural. And what a granum is essentially a thylakoid membrane. That's one of these things. So this internal membrane structure houses the proteins that are going to facilitate the light-dependent reaction. So a granum is essentially a thylakoid membrane wrapped up, and it has a space in the middle. Now, if we kind of looked really closely at one of these, one of these granum, and we looked at the structure of it, here is one of those granum enlarged. So this is just basically one long thylakoid membrane. Along the membrane, we've got transport proteins, and we also have structures called photosystems. So there are two photosystems, as we'll learn about as we look at the next reaction in a second. Photosystem 1 and photosystem 2. Now this is partly to do with when these were discovered, but it's also to do the naming of them, to do with the wavelength of light that it deals with. So what basically happens is, we start off with the water. The water is struck by light. That splits into electrons and protons, as we're going to see, and the electrons are carried down these transport proteins on the thylakoid membrane within the granum, and as that moves, it generates ATP, and then those electrons move and are carried from one photosystem to another, and really vice versa. I should be putting photosystem 2 because that's the one that appears first, and then photosystem 1. So we go from one photosystem to another, and then it moves down another carrier system, and then we eventually end up with the products that are then going to leave the stack, the granum, and go to the stroma where the next reaction is going to take place, which we'll see in the next video. So let's draw this out and talk about it step by step, just to give you the overall picture there that you can refer back to. So what's going to happen is, the first stage is that a molecule of water is broken down in a reaction called photolysis. Photolysis, photo meaning light, lysis meaning to split, splits the water molecule up into oxygen, which is going to leave out of the stroma. So there we immediately have used the water and produced the oxygen that's taken place. Now, the oxygen is one of the products there, and we also have the hydrogen atoms being split into electrons and protons. We're going to make 2 protons and 2 electrons. And what's then going to happen is these electrons are going to be passed into this electron transport chain. So we've had light energy strike the water molecule and split this apart. Now, these electrons are then going to be moved and transported to one of these photosystems that are present in the thylakoid membrane of the granum. And this is the area we're talking about here. So the first photosystem is photosystem 2. Now, what do I mean when I keep saying photosystem? A photosystem is just basically a protein complex that contains lots of different photosynthetic pigments. Now, At GCSE, we say this is chlorophyll, but the reality is that different plants absorb light at different wavelengths. So we might have photosynthetic pigments that absorb green light or red light, depending on the plant species and what it's adapted to use. So this photosynthetic pigment is present in the photosystem, and as the light strikes the leaf and strikes the chloroplast and then moves around within the granum, eventually it strikes the photosystem. And the electron that we just passed into there, because it is struck by light energy, is excited to a higher energy level. So as we go higher on this diagram, we're looking at change in energy here. So we're going to have more energy. So obviously it absorbs energy, it becomes excited, is the term we say. So what then happens is this electron is passed onto a transport chain. Now, in this transport chain, the electron is going to move from one protein to the next. You notice the proteins are gradually lower down because each one works at a slightly lower energy level. And that difference in energy is used to generate ATP by a process called chemiosmosis. And we're going to learn more about this in the respiration video, which is going to follow subsequently. So just for now, if you accept that because the electron is being passed down, ATP is being generated at this stage. Now, that electron is going to continue to move down this chain, as we mentioned here. And so it's gone from photosystem 2 to the electron chain, and now it's going to go along another electron chain, and it's going to arrive at photosystem 1. So here is photosystem 1. Within this photosystem 1, we've got more photosynthetic pigments. The light is going to strike the electron that's being housed within this protein complex, and that's going to excite it. And when it becomes excited, it's accepted by another electron transport protein. And that's part of a chain, and the same process takes place, this time without the generation of ATP. But what does happen here is that the final molecule is a coenzyme called NADP. Now, a coenzyme is just a molecule that enables enzymatic reactions to take place, or it helps with the reaction. And when this coenzyme NADP accepts an electron, it becomes reduced. Now, you might remember from GCSE, we talk about oxidation and reduction a little bit. Oxidation is loss of electrons. Reduction is gain of electrons. So when the NADP becomes reduced, it becomes negatively charged. And as a result of being negatively charged, the protons, and we'll talk more about the role of protons in chemiosmosis, the protons will combine with the NADP to form NADPH. Now, if we say NADPH, we see NADP minus, we're still talking about reduced NADP. It's just a NADP coenzyme that's gained an electron. Now, the reason we sometimes see this as NADPH2 is because we made 2 electrons within photolysis. So this whole process happens again. Another electron goes down the chain and goes all the way to the end and combines with the negative NADP, and then another proton combines to form NADPH2. But essentially in all the textbooks literature that you'll see, we're referring to a reduced coenzyme. So that leaves us 2 products here, reduced NADP and ATP, that have been formed within this chain. We've excited the electron once here, it's gone down a transport chain here. We've then arrived at another photosystem and excited it again with another photon of light, and then we've gone down a transport chain and finally reduced this final acceptor molecule here. It's reduced NADP. Now, what's the difference between photosystem 1 and 2? Well, you'll notice that this photosystem is at a slightly lower energy to this photosystem. So this works with slightly higher energy wavelengths. Even though the electron has lost energy as it moves down here, it still has more energy when it's at this stage than it is at this stage. So the reason we say photosystem 2, it is to do with when they were discovered, but we also discovered this second photosystem that works at a slightly higher energy level, so we depicted it as being number 1, or first position, first place. That's the way I like to remember it. So this whole thing is a linear reaction. It's not a cycle like the next reaction we're going to see. So we say this is non-linear cyclic photophosphorylation. Now, phosphorylation reactions are reactions where phosphates are added to ADP generally. Can be other things, other molecules to make them more reactive, enzymes and such. But generally when we're talking phosphorylation in A level, we're talking about phosphorylation of ADP to make ATP more commonly than not. So we've not got a cyclic reaction, it's non-cyclic. We've used light at 3 different stages here, and we've phosphorylated ADP. So it's non-cyclic photophosphorylation. Now those products that have been made within the thylakoid membranes, within the granum, are going to go from this area into the stroma, where the light independent reaction is going to take place.